I cut off earlier. Wei-Jun?
Wei-Jun Tsai, University of Delaware. So my comment and question goes to the second and third talk this morning. You guys, compare that, what did I call it, SIR and the carbonate. Which one really, is this working? Yeah, which one really is responsible for correlate the carbonate dissolution? I felt that's both fascinating and laughable. Sorry for saying that. But I'm pretty sure you look at the thermodynamics, so the dissociation of second step K2 exactly linked both. So the ratio of bicarbonate divided by proton multiplies K2 equals carbonate. So when temperature salinity is the same, then both end side of the equation are the same. So only when temperature changes, K2 changes. When you warm up, K2 increase that, increase carbonate. So I think if you could go back to those cases, they agree. Are they constant temperature case? But those cases they do not agree. Are they because of that changes? So that would be a good direction to look into. Thank you for your.
Thanks, Wei-Jun. I'm wondering if maybe it just didn't come through, or I'm not entirely understanding the question, but that was our attempt to take out the thermal effect of the biological response. And so we had, and that's why we ended up with only 7 studies at that point that had actually varied the temperature in a way that wasn't harmful, where we could get the SIR to vary at a constant, or nearly constant saturation state. And so in doing that, then correcting those biological rates for the thermal response, then we fit those correlations between the SIR and saturation state, and effectively show that in general we can explain more variance with the saturation state than the SIR. And again, I fully recognize that they're not completely separate either, right? And even in the context of if we're to the point of saying that SIR largely is a function of the proton concentration, having more carbonate there buffers protons as well, right? So there's some inherent circular arguments through the thermodynamics that we can't entirely separate, but we can try to get a sense of empirically what actually seems to be a better predictor of calcification.
When temperature is the same, both sides are exactly
exactly, and that's the fundamental problem, right? And that's the problem with the fact that we don't do experiments at intermediate treatments often because they're complicated and they require a lot of work. So we often pick 2 end members and hit things with hammers and say they're sensitive to temperature or they're not, or they're sensitive to acidification or they're not. And so being able to fill in those intermediate kind of values gives us an opportunity to do that.
Yes, I agree. I mean, it is virtually impossible to separate the influence of both of them. So I would argue that SIR is like perhaps more relevant for the calcium. I think we have to distinguish production of calcium carbonate of dissolution, right? And what we measure in experiments is in most cases just the net result, right? So we can have influence of both in the end, right? I would argue that this ratio is perhaps the predictor for production, whereas omega is, of course, for dissolution, the relevant factor. Yeah, and I think you can only, physiological understand it when you really truly understand which inorganic carbon substrate is the source of calcification. So is it bicarbonate or carbonate? And yeah, so it's very difficult. So I dig through the physiological literature, and there is no, also not in the medicine literature, there's no study that says, okay, our bicarbonate transporters on a molecular level transport bicarbonate or carbonate. They just simply cannot distinguish that because the interconversion rate is extremely quickly, right? So that is our problem where we're standing at. So we can mostly argue. And then George's approach, of course, to change the temperature. And I think it's a great way, but I'm just a bit careful because temperature has such a high influence on calcification as such that when you try to detangle it with the temperature component, that you're never sure if you get rid of that confounding factor entirely. Right? So that's my opinion.
Julie Granger, University of Connecticut. So I may have missed some details, but in comparing Wei-Fu's talk to George's talk, Wei-Fu treats the physiology as a compartment that doesn't talk to the calcifying component of that other compartment, right? Because people asked you about photosynthesis and the respiration of the organism, and you mentioned, well, your model, they're two different components, so it might affect the external pool of pH, but there is a membrane that, that could potentially connect the physiology to the calcifying space, whereas explicitly you mentioned how the physiology affects the calcifying space. So is there a disconnect, or I just missed something entirely?
I guess I could start. I mean, part of it is where the tissue is relative to the calcium carbonate relative to the environment, right? So, and I won't comment much on the corals because it's not really my area, but for the bivalves anyway, they have this tissue that sits inside the shell. The outside of the shell is partially protected from the environment from an organic layer that's on there that often gets damaged in the environment. But the inside of that shell is where they actually calcify. And the attachment of the mantle or the tissue to the shell actually varies quite a bit amongst different bivalve species as well. So if you think about a mussel, if you've ever eaten a mussel, they're kind of stuck to that shell pretty well, and an oyster is pretty free. So the ability to isolate that space is quite variable. And we see that because think about, the other example or extreme is a land snail that calcifies, right? They're using 100% respired carbon as their carbon source for the calcium carbonate. So we don't see that in the bivalves. The largest numbers I've seen in literature using stable isotopes suggest 30 to 40% of the carbon in the shell is respired carbon. And then you can even look at the inner space versus outer spaces, and there's been some interesting bacteriological studies that show in clams the inner calcifying fluid is more different bacteriologically from the outer fluid, which is more similar to the outside environment. So I think that's the mollusk perspective, I guess. But they're quite, different in terms of where all these layers are situated relative to a coral.
So I just want to clarify what I said earlier this morning. What I meant is not the calcifying space, not communicating with outside. What I say is that interaction is embedded in the model as a parameter like a carbon transport. So that's kind of like a free parameters. If you think they are communicating more carbon from outside to inside, you increase that parameter. Similar thing as a pumping, that might depend on what's the proton concentration outside. So I guess my main point is there is a mechanism in this model you can take into account. But what I found in my model, when I try to compare my model output with experimental data, it shows when you grow coral at a different seawater pH, that parameter doesn't really change, which means the coral, this kind of interaction doesn't change that much, even you change external seawater conditions. Does that make sense?
I'm just wondering how much photosynthesis and respiration change in those experiments, or if those are constant, then you could have an effect that you wouldn't see, but I didn't understand what was constant in the experiments and what wasn't.
Yeah, so there has been experiments have done trying to characterize what's the photosynthesis activity and those kind of things. So what do we find? In some cases, it also depends on whether you run short-term experiment or long-term experiment. For short experiment, you sometimes see a very big change in those parameters. You'll see if I want to fit my model to those kind of experiment, you need to change like a pumping rate or carbon transport rate to make the pH matches. But in the long-term experiment, you don't really see it. So it kind of depends. I think that's the part of the biology is so fascinating. They can change on different time scales. They can also do something crazy you don't understand, especially when you do some treatment to the coral. They can basically go crazy for a while, then after a long time, they settle down and behave normal. And the model basically provides a way to gauge whether they change or not by fitting the model to the data.
Hi, Yui Takeshita.
Introduce yourself.
On top of it. Yeah, before I was rudely cut off. I am Yui Takeshita from Embark. This is actually a really nice segue into my question is that on coral reefs, there's really like high frequency variability, where pH can change by tenths of a unit on tidal to diurnal cycles. And so my question to you is what's your, so in terms of time scales, what's your time constant of your model? So if coral, so that you showed very nice seasonal agreement, but then if pH is changing by that much on hourly time scales, can the biology change rate constants that fast, or do they kind of see an average? And then so, because there are more and more data sets that look at hourly time scale for days to weeks, and then, but can this model be applied? And kind of similar to bivalves too, like, in those 6 hours, if, depending on what phase of tide you are, you could be seeing a completely different pH. And then so kind of tying that laboratory experiment to the field where you have to interpret in the context of natural variability, how do you guys think about that high frequency variability, which is really important to interpret all this?
So I can start. So for coral case, in terms of model, what I presented, the time scale is very fast. So the resident time in my model for the calcifying fluid is on seconds. So if you make a change, you will rapidly see what's going on in the calcifying fluid. And also in the lab experiment, people have tried to use microelectrode, pH-sensitive dyes. They see very dynamic change of pH on minutes or hours. So it's a very high frequency change. But what matters in long term is how you integrate it is. Because some of variabilities can be the physiology suddenly change a little bit, coral calcifying fluid will respond. But in the long term, it's the seawater chemistry controlling it, both on the short time scale and long time scale. That's what I think. Then it becomes a problem. You basically have a pH variation, as you said, on diurnal or seasonal scale changing. Then you integrate the pH signal over years, over maybe decades. What's the accumulative change in pH or calcifying fluid pH change?
So your model, it is applicable on minutes to hour scales?
Yes. If you know what's the seawater change on those time scales, yes.
So, and just from the bivalve perspective, I think it's important to remember the sensitivity we find is really in the larvae. We do see changes in calcification in adults. That's not going to probably kill them. Over integrated enough insults over time, maybe it results in lower reproductive output. But I wanted to point out my graduate student, if you could, or former graduate student, successful graduate student, Idia Jimenez, recently successful. She's got a really nice paper. We took an approach because you're right, in the environment, these things, especially in estuaries where oysters and bivalves are found, is quite variable. And so we did a number of experiments where we allowed conditions to vary or not, and developed essentially just an integration. It's similar to a degree hour, degree day type perspective, and it's a degree acidification perspective. And what we had to do was heavily weight that initial period and found a fairly good predictability of survivorship of larvae by sort of integrating over time in that exposure window. And what we found in those experiments is if you didn't expose them during that initial critical 6-hour window, the survivorship was much higher than if you did expose them and then move them to a stable, constant condition. You still saw a lot more survivorship problems. So it's a really complicated issue, and I think, in many ways, the EPOCA group tried to sort of consolidate and get everybody to think about doing things a very specific way, but it turns out that once we started really thinking about this problem in estuaries, that was not really appropriate for how we sort of do experiments, and it's really hard to do these experiments and do exposures that vary over time in some way that you can control them. So I think it's an area we're still working on, trying to do better.
Chris Sabine, Hawaii. Given the questions that have been raised today and the level of interest in the room, I guess I have to ask the obvious question to Leonard. Why would you say that OA research is done?
Yeah. Okay, I guess perhaps two parts to the answer. I mean, the session reflects it a bit as well. I mean, we're talking about calcification, and we only see it in the carbonate chemistry context, right? I mean, there is so many other factors that affect calcification, and by the dominance of this topic, we tend to oversee those. That's, I think, a big problem. And the second thing why I think this path, this research direction is, for me at least, not the way to go, is because it's a political answer, actually. I don't think that in 10 years from now, when I continue ocean acidification research, that we have come to the point that we significantly better understand. I mean, maybe we do some progress, right? But I think in 10 years, we have a limited carbon budget, and in 10 years, we have expired most of that budget already. So in the context of the global developments, I think we have to move on and work of solutions for climate change and not necessarily on the consequences. Because I think we have actually convinced the public already. I mean, at least in Germany, that's what I see. People are on the street, right?
Have you heard from our president recently?
I know, but I mean, I'm an optimistic person, and I think that, I mean, even in the more right-wing, okay, that's getting too political. But anyways, I think they are reasonable, thinking people, and yeah, I think at some point it's just becoming too obvious that this path of sustainability is more attractive than the other path.
Jim Bishop, UC Berkeley. This is a follow-on from your last bullet point on your final slide, which was geoengineering. Could you elaborate on that a little bit about how calcification feeds into geoengineering solutions?
Yeah, so I mean, geoengineering, or that term is actually burned pretty much, so we tend to give it another name, but I think we can stick to that term. I mean, it's now, from the IPCC, it's very clear that this is an essential tool to achieve the 2-degree goal, right? So we can either say we want the 2-degree goal plus geoengineering, or we can say, okay, forget about the 2-degree goal. There is not much room in between. So I think, I guess as a scientific community, I don't know, is Andreas still here? Andreas Oschwitz? He's having that priority program on that already. And I think, yeah, so for us it's now to anticipate that and understand the risks associated with that for the ocean environment. So that's why I was saying I think this is the new ocean acidification topic, that we have to assess the risks of that pathway, right? Because we know the risks of ocean acidification already. And I really seriously doubt that in 10 years from now, we say something completely different that we say today.
Hi, Pat Rafter, UC Irvine. My question was also for Leonard, but I'm gonna take a 180 and say, Kristen, can you comment on Leonard's statement that E. huxleyi is not the most important coccolith in the ocean?
Yeah, I think that the other ones are obviously really important. The subtropics and tropics are mainly inhabited by other species. I mean, there are E. huxleyi everywhere, so that is a really important one to understand. But it's also the most experimented with already. And so, if you looked at the number of dots on my meta-analysis of all the physiological studies, most of them were E. huxleyi because that's what people use to study. And I think that was okay for building this parameterization because there is E. huxleyi is really widespread, and so that's kind of appropriate that it should be the relationship should be biased towards that. But it is true that you know, if E. huxleyi doesn't do so well, then other ones could take over, or vice versa. So I think that in terms of Leonard's statement, I mean, I don't necessarily disagree because there's already been a lot of studies on it, and so we already have that data. And then just understanding what the other species are going to do is also really important.
Hi, Matthias, UBC Canada. I will take debate on geoengineering and thinking about, for example, iron fertilization, which targets diatoms, and thinking in the long term. Those, if they're used for carbon sequestration, they would sink and take with them all the nutrients. In contrast, coccoliths are mostly carbon, and if they're used in a geoengineering context, they would be, I guess, less harmful for the inorganic stock already in place. In that context, what do you think is, in the long term, the position of the ocean as a carbon storage place to put all the 415 ppm in the atmosphere now? Because, well, we have to think about this.
Anybody want to touch that one?
Well, I'm not quite 100% sure I understood the question. You're saying that is the ocean the place to store all that carbon? Well, there isn't much choice, is there? Okay, so we have to find a means. If we're storing carbon, it's the biggest reservoir we can put it into, all right? And do the least amount of harm in some ways. We just have to figure out a way to do that, and then do that in an economical, ecological way. That's the way.
So I actually, for this room specifically, I want to walk that question back just a little bit and ask each of the panelists as a way of rounding out our session. You know, you all talked about changes that are happening to calcification, whether it's to particular organisms or species groups, population diversity, calcium carbonate compensation, et cetera. Where do we need to be working? What process studies do we need to run? What regions do we need to be monitoring to keep track of these changes so that we can better understand how things are going to change in the future and help build resilience in a variety of different ways?
So I think the only thing I'd really, I dig bivalves a lot, so obviously we need to study and understand them more. But from an engineering, climate engineering, or however you want to phrase it, there's a lot of interest in trying to do things on local scales that don't require the global buy-in or the massive scaling operation. It doesn't solve the global carbon problem, but it helps to maybe buy some time. And so people are looking at phytoremediation, looking at ways to use carbonate at local levels to offset the experience of organisms on these smaller time and space scales. And I think that's an area where there's a lot of work ongoing. And I would sort of frame that in the context of it's not a blue carbon perspective, but rather a chemistry mitigation sort of perspective, in that we don't expect we're going to store tremendous amounts of carbon in any of these systems if we plant more seagrass or restore oyster reefs to their once former glory. But they might help alleviate the experience of organisms in those systems and buy us some time while we figure out how to do this on a much broader scale.
Kristen, where do we work?
I thought that, okay, so according to my results, I would be most interested in seeing some coccolith-4 change studies in areas where I saw the biggest increases in coccolith-4 biomass, such as the Patagonian shelf, or we already have some observations from the North Atlantic, but also the North Pacific, and also some of the big areas where we saw the biggest decreases in calcification, such as the Southern Ocean or even the Arctic, but that's quite hard. And the coccolith-4s don't generally grow too much up there, but they might migrate up there considering climate change.
I'm so biased. I heard the word Arctic and it made me excited. Bernie, where do we work?
Well, we want to store it permanently, as far as humans are concerned, right? And the best place to store it would be to react it with the bottom. Make the bicarbonate down there. How do we do that? What are the energetics involved in that sort of thing? I don't have an answer. If there was an easy answer, someone would have come up with it a long time ago.
Andrea.
I don't know what the question is anymore, to be honest.
To bring the room back, we're talking about
Answer what you like.
Seen a lot of discussions of ongoing change in regards to the calcium carbonate cycle. What do we need to monitor? What process studies should we conduct? Where do we work to monitor these kinds of changes?
Yeah, I mean, I don't know the answer to that, but I've been thinking a lot about the seasonal cycle of phenology changes, and so that's one of the reasons I was so interested in this session in the first place is, you know, some of the nitty-gritty about what drives calcification might seem overly, I don't know, narrow, but I think that if you know, those drivers are important in the long run, it's important to get them right based on Bernie's talk, which is slightly horrifying. But of course, you know, there's also, Leonard I thought you did a really good job presenting the point about other stressors really influencing our interpretations of calcification responses. And if we really need more ecological understanding to do better in models, that makes it even harder to get the data we need. So one thing I thought about was, Kristen, you have a model. You can actually change some of these sensitivities and look at the range of variation and see what put a number on the magnitudes that we might be looking at for variability.
Leonard, Wei-Fu, where do we work?
Yeah, so from a purely scientific point of view, I think the challenge will be to make the right link between ecology and biogeochemistry. So how do ecological processes shape the calcium carbonate cycle in the ocean? Because I think we have done bottle experiments like crazy, but as I said, there's more than, the ocean is not a bottle, right? So I think we have to expand on that.
Yeah, I agree with Leonard who said. I kind of touched on this in my talk. And maybe it's for my ignorance, I don't know much what's controlling the reef carbon chemistry. So I think that's one area. If I can learn from you or from any expertise, it will help a lot. And I also agree with Andrew just mentioned the multi-stressor problem we are facing. As I show in my talk, calcification have two components, one extension and one densification. It seems like we have a better understanding about densification, but extension, what's controlling extension is not the carbon chemistry. It's the temperature, it's nutrients, what's controlling that, because that's a big player. When you're looking at the coral call, the biggest variation is coming from extension. So we need to solve those kind of puzzles in order to make a complete, accurate prediction of what will happen for coral or for other biocalcifiers.
So I'm standing between you and lunch. I want to thank our speakers one more time. And during lunch, ask your colleagues about the calcification mini session that happened before OCB. I'm sure they have a lot of clear insights to share.